you so much, Philippe, for the introduction. So yes, for the ones that don't know me, I did my PhD here in the group 2 years and a half ago. And then I moved to the Netherlands for a postdoctoral position in the Netherlands Cancer Institute in the computational pathology group. And since then, I've been working mostly with what I will talk today, that is approaches to combine H&E and immuno, and I will tell you all about this. But it's basically just a way of trying to combine these two types of images that we have in pathology. But before going into a lot of details on the technology, let me start with, oops, sorry, this is not working as it should. Okay, let me start with the very basics. That is what is pathology. Basically, this is the study of thin tissue sections of tissue that are collected from tumor samples. And then pathologists just want to assess the morphological features, then they can use to identify the disease subtype, the progression of the tumor, and the severity. And this is super important for a detailed diagnosis and then the treatment management. And with the traditional workflow, what happens is that they collect the tissue samples with a biopsy or even surgery. Sometimes it's the case. These pieces of tissues are then prepared and mounted in a glass slide and then stained with special stainings. The most common one, or the standard one, is methoxylin and eosin that gives this purplish or pinkish tone to the samples. And then these slides can be analyzed by the pathologists under a microscope. With the transition to the digital workflow, now we have scanners. And so these tissue samples are now digitized, and the pathologists can have high-resolution images. And so they don't have to see the samples under the microscope, but they can have the images on a screen. And these type of images are called all-slide images. And what are the details of these kind of images? So these are really detailed and really big images of the tissue. This is just a representation of the entire glass slide. And they are digitized at the same resolution as the microscope. So we have a very fine level of detail. And they also have these pyramid formats that basically mimics the microscope lenses. So basically, the lower the level, the higher the resolution. And when we go up, it just like becomes smaller, and so we see more. So we have a lower dimension, but it's exactly like the pathologists see in the microscope. And so for you to have an idea, we can go from low magnification, and we have like an overview of the tissue, and then we can go up, up, up. And then we have a lot of resolution. Sorry. This is not cooperating, but I want you to see this image. So we can go to this level of detail. We can see the individual cells at a really high level. And now with all this information being available as digital images, of course, this just open this big area of computer vision and AI applied to pathology. And then we have computational pathology. And basically, this is like the main goal is just to design learning models for whatever task they might have in the pathology lab. As for example, automating cell counting that pathologists sometimes have to do to have some scorings, do tissue segmentations so they know where the tumor is, for example, or for example, to perform some artifact detections that can occur at every step of the workflow, or even like maybe with a high-end goal that is like predict return responses or even discover new biomarkers. And these are just some examples for you to have an idea. And what do our images look like? Well, basically like this. You have pink images, then the blue, and then these black and really bright images. Starting with the methoxylin. So basically, this is, as I said before, this is the standard staining that we have in the lab. And what happens is that they use a combination of these two reagents, that is the methoxylin and eosin. And the methoxylin highlights the nuclei because it binds to the DNA, so it turns the nuclei purple. And the other one binds to other proteins in the cells, and so it dyes all the other structures in pink, and so we have this contrast. And then we have immunohistochemistry, that is a more advanced technique, and here they use antibodies to highlight some specific proteins within the cells that then appear as brown. And then we have multiplex, that is basically even more advanced immunohistochemistry technique that uses fluorescence and enables also the visualization of several markers, but at the same time, because each marker has a color, so they can put all them together, and then you have you can distinguish all types of cell with only one sample. Even though we also have work in multiplex, this is not really used in the clinical workflow, it's more for research. And so today, and also my research line is way more focused on methoxylin and immunohistochemistry. And today I will be focusing on these two. And my topic is a bit out of the box, if I could say. That is, instead of just performing any other task in one of these two modalities, we were wondering if we can go from pink to blue. And so from one domain to the other. And this can be done, or please believe me, this can be done with virtual staining. And this, I started this research line last year with a project that is funded by the Dutch Research Council, or the Dutch FCT. And it was a partnership with FIOP and the lab, pathology lab here in Matosinhos. And basically, in this project, we were focusing on breast cancer because it's still the most incident type of cancer in women. And these can have like multiple subtypes of disease, let's say, and also then severity. And these subtypes are diagnosed based on several biomarkers, several immunohistochemistry biomarkers. So basically, the pathologist just evaluates the presence of certain hormone receptors, for example, the estrogen or the progesterone, or for example, other markers like the HER2, that is the one that we are interested in. And basically, HER2 is like a membrane in the protein of the protein in the membrane of the cells that plays a role in the development and the progression of the cell growth. And so some of the most aggressive types of breast cancer have these protein very developed, or they have a lot of these receptors in the membrane, or some other, so the cells just grow a lot more. And some others don't have it all. And so it just makes it more difficult for example, immunotherapies that target this kind of this type of receptors to be less efficient. So it's very important to understand if these markers are present or not. Also to decide treatments. And this marker is basically scored in 4 categories. And the higher the score, the more intense the staining, so the more brown it appears on the images, and then the more severe the case, let's say. And however, what is the concept behind the virtual staining and why? Even though these markers are routinely used in the lab and are very important to diagnose the different type of breast cancer, in the normal workflow, first the lab produces the H&E slides, and then the pathologist evaluate these slides, and then they request the immuno if necessary. It might be a bit different. The immuno might be available already, but most of the times this is a process. So it takes time and it takes money because it takes another sample to be prepared. And also, when this is requested, again, this new test is performed in another sample of the tissue. So imagine that you have the tumor, they just cut thin slices. So first they cut the first to do the H&E, and then they have to cut some more and cut another to do the immuno. So when the pathologists are assessing either both the immuno and the H&E, even though they belong to the same tissue sample, the same tumor sample, they are not seeing the exact same cells in both. So there might be some mismatch, or for sure there's no the same cells in both. And so our goal is basically to generate these second slides, the immuno slides, without the need of any antibodies, and then simulating the staining on the screen from the H&E directly. And why would this beneficial? So we have 2 points of views. First, from the lab point of view, if we are able to virtually mimic the staining process, basically we can generate multiple digital slides of whatever marker we want. Now we are focusing on the HER2, but this can be applied to any other marker. And we can do this from a single cut and a single sample stained with the H&E. And so basically we can preserve the tissue, the tumor tissue. Of course, we reduce the lab workload and also the costs. It just makes everything more efficient and also eco-friendly, because then we don't need the antibodies that are first super expensive and then also not that eco-friendly. And then from the pathologist point of view, or the clinical point of view, this can be used as an indication of the immuno without having to perform the immuno. It also reduced the turnaround time, right? Because if they have the H&E, they can just like push a button and then they have the H&E result, and they don't have to ask for it. It also allows to customize the staining patterns, because imagine that I have that 4-marker panel that I showed before. From one H&E, I can decide which marker I want to see, or if I want to combine all of them, and I will have all of them in the same piece of tissue. And exactly, we have everything on the same cells, so we don't have this mismatch that usually occurs. Okay, but how can we do this? So basically the goal is to map one image domain, in this case the H&E or the pink images, to another image domain that is our target, that is the immuno. And basically there are 2 types of models that we can use for this task. First, we have the usual suspects, that are the GANs. And more recently, we have the diffusion models. And even though we have explored both approaches, I had, like José, that is a master student that I supervise with Professor Jaime, and Pascal, that is a researcher at the NKI, working with me in this project. And even though we tried both, today I want to focus on the GANs. It was the work developed by José for his master thesis. And let me start this part with showing you what is out there already for this specific task of virtual staining for HER2. So basically there are 3 works that we can consider as like the reference for this. And first is the pyramid peaks to peaks, and it was introduced by Liu et al. In 2022. And this is basically the foundation of all other models that were developed. Okay, all the publications are somehow based on this. And here what they did was just they introduced this concept of having a pyramid loss. So exactly the same way as we have the pyramid and the different levels in the old slides, here they do the same. They just compute the loss at different levels. And then here, after this, Lee et al. In 2023, they just extended the pyramid peaks to peaks, adding a contrastive learning objective that they call adaptive supervised patch NC. So that's why it is called ASP. And then we have the third, that is PSP stain by Shen et al. And this, again, extends the ASP, but here they try to incorporate the pathological consistency loss, trying to compensate or trying to enhance the staining, the biomarker expression. So the way the brown appears in the image. And as a first step, before delving into some model development, we just evaluated these three models. And we use not only the standard metrics that are usually used for image evaluation, that is the SSIM or the PSNR, but we also use other metrics that are a bit more aligned with how we perceive the images. That is the LPIPS or FID. And basically the difference is that SSIM and PSNR just evaluate the image at the generated and the real ones at the pixel level. With the LPIPS and the FID, these similarities measured at the activations or the final embeddings. And so this evaluation just differs in terms of what is like the exact pixel look like or what the global images look like to us. Also, we compute another distance, but this time only at the, so basically we have two colors here. We have the blue and we have the brown. The blue is still amatoxylin, the brown is DAB. It's just precipitate, so a molecule. And so the brown is where you can check the marker expression. And so this distance is basically what you want to have represented in the image correctly for the marker evaluation, of course. And so we basically just evaluate the distance between the original and the generated image at this level. From the results, what we can see from the table is that the pyramid peaks to peaks and the ASP have the highest SSIM and PSNR. But then if you look to the images, I hope at home you can see it a bit better. They don't perform that well, right? Even though they have these metrics a bit higher, then in reality, the images don't look that good. And in contrast, we have the PSP stain that has like a better JST, so the distance at the dot, and it really aligns with the images. So it seems that it has a better biomarker representation, but then it scores lower on traditional pixel level metrics. So what we concluded here is that basically these traditional metrics are not the best to evaluate such images because we can have really good values and even like models that seem to perform really well at this level. But then when we compare side to side, this is not the case. If we look to the images in detail, for example, the pyramid peaks to peaks, it's very like smooth and there's no cells at all. It's just a blur. And then even though the other two perform a bit worse, or even like the PSP stain, that is the one that looks better on the images, they have like really worse SSIM or PSNR, even though performing better at the image similarity. And so basically, this is the first point that I would like to raise. That is a problem with the metrics and how to assess the quality of these models and the correspondence from the generated to the real images. In terms of data, there's two datasets that are publicly available, and they come from the two first works that I talked about, the pyramid peaks-to-peaks and the ASP. And so these are the VCI and the MIST. They are quite similar in terms of size, in terms of features. And they are also one really important detail is that they are basically consecutive slides. They are just slides retrieved from the archive. So they are slides that the pathologist, the lab performs as normal. So basically, they correspond to different tissue cuts. Okay, even though they belong to the same tumor sample, they are not the exact. They don't correspond exactly. More than that, when we were doing this evaluation, and also when we started developing the model, we start wondering, like, but why does our model doesn't perform that well? We are kind of doing everything. And then we checked the data a bit better. And what we found was a lot of artifacts, as, for example, the tiles not having enough tissue. So it's basically background. Of course, if you assess the metrics here, it will be super good because it's all white, right? Even though the model might not be learning anything. We have general artifacts, like, yeah, there in the screen, you can see that well, but basically because they had to register the two slides, what happened is that there's a defect on the tissue, kind of a brush sometimes. And so we have we see like a stripe, so like some repetition, or even worse, that is like complete distortions on the tissue, like on the right. And this was, we first assessed the test sets. And in the training set, there's also two other problems, that is not corresponding tiles at all. So for example, having just background on the H&E, but tissue on the immuno. Or for example, having just blank tiles, so nothing. And more, like, I think it's even worse when it comes to numbers. So only 29% more or less of the training tiles and 15 of the test are kind of usable. This doesn't mean that they don't have any artifact. They might still have some small artifacts, but at least it's not one of these. So they are usable. But as you can imagine, when we conclude this, we were quite amazed because all of the models that are out there are based on this, are evaluated on this, are trained with this. And so, of course, it's just a limitation, right? And together with the limitation on the metrics that I told you, having these data sets that first don't have the exact correspondence, plus having the artifacts, with this comparison being done with like normal metrics that are not that well adjusted to these tasks, plus the models trying to put a lot of losses to trying to compensate for all these errors. This is not the, let's say, the best. And so our approach was basically taking a step back and trying to understand exactly where we can improve. So let's start with the data. In this project, we developed a new data set that we released publicly, so everyone can use. And the difference of these is that instead of having just consecutive slides, what we did, we took the H&E, we removed the staining, we did the staining on top, and then we have the exact same sample with the immuno. And so when we cut the tiles, we have exact correspondence of the cells in both type of images, right? And this was done by Pascal and curated by him and me. And in the end, we have a data set that is, well, it has a higher resolution than the previous ones and have because of this, also a bit more data in terms of number of tiles. And then in terms of the model, so as I said before, I will focus on the GANs, our proposal for a GAN. And so the first goal, and this came from our evaluation of the state of the art, was to explore the potential of the discriminator. So far, as I said, all the authors were really focused on topping losses, like putting losses on top of each other, and not really kind of taking advantage of the structure of the GAN. So basically, they were not exactly using the discriminator that much. And so what we did was explore that exact capability. And as a first step, we just concatenate the H&E and the generated immuno to pass to the discriminator, kind of to enforce the preservation of the semantic information that we want to preserve from the immuno. And why? Because there's a big assumption behind all this work. That is, there's information on the morphology of the cells that is assessed on the H&E that can be used to then infer the biomarker expression. And so if we concatenate this, is a way of trying to leverage the adversarial loss, not only to produce like a real look, like realistic images, but also to preserve the pathological meaning or semantics. On top of these, we extended, and then we just used the binary cross entropy, so very simple here. And then on top of that, we added the CSS loss. And so basically the idea here is to look at the structure of the staining, like more the membrane, because this is the membrane staining. So it really highlights the membranes of the cells. And so we were trying to focus here and these really local differences. And then we want to penalize structural differences from the H&E and the generated immuno, because we want, like, the cells are there. So we want to preserve the information on the cells and just trying to synthesize the staining, right? And this comes from this idea that when we compute the CSS index, so the structural similarity between the H&E and the immuno, we end up with this kind of map, where the dark regions are the ones where there's like a bigger difference on structure, or they don't share the same structure, and so they are more penalized. And then lastly, we just retrieved the idea of the pyramid to try to penalize the differences between the generated and the real immunos. Sorry, there's an error here. This is not on the H&E, it's just the generated and the real immuno. And here it's just like trying to improve the similarity of the generated and the real immunos at different levels. Okay, and so it's very slow. When we compare all of these variations, basically, even though we came up with the CSS loss to try to improve the preservation of like the H&E or the cell structure, what we concluded is that the main contribution is the training regularization, because then the DAB representation is really improved. This is very difficult for you to see here on such small images. But the thing is that because we are trying to account for the differences at the structure, so it's like luminance, contrast, it really highlights the difference in, like, when it comes to the brown, because it's the most different for of the stain of the H&E. Also, we see a little improve or a small improvement in LPIPs, even though it has a decrease on the general metrics. But again, in the same trend, the general metrics don't really correlate that well with the results that we see in the end. And then adding the pyramid peaks to peaks just improves these human perception metrics, so the LPIPs. And then we have like improved performance when we compare with the very first step of the model. And of course, we then have to compare with, let's say, state of the art. And because our model kind of combines a bit of the pyramid peaks to peaks and the ASP, these were the models that we compared to. And so we retrain both models in our datasets. And from the table, we see that the ASP achieved the top performance on the traditional metrics, so SSIM and PSNR. And then in terms of the MS SSIM, this is just, again, the SSIM applied to different levels. So it's a multi-scale SSIM. And for these, we have the pyramid peaks to peaks as the best performing model. But then, if you look at the images, here they are. And we have the exact same trend as before. Our model is the last column. And I mean, I think it speaks by itself, right? Even though the other models have better metrics, they don't have better images. And there's one tiny detail that also might partially explain the difference in the not so good metrics, is that even though our staining is performed on top of the H&E, let's say on the same sample, in the lab process, there might be some tissue loss, just because, so we have the glass slide and we have a cover. If we have to take the cover, it just sometimes takes a bit of the tissue along with it. So of course, there might be some tissue loss and some like pixel misalignments. So of course, even though this is much closer to complete correspondence, it's not 100% perfect. So of course, all the metrics that are pixel related will be affected by all these differences. But okay, but more than the metrics, we decided to do a qualitative test. And we asked 3 pathologists to evaluate 500 randomly selected images from our test set. And these images are also spread across those first scores that I showed, from the less stained to the more stained. And then they had to, so they were seeing the images side by side, the real one and the one generated by our model. And we asked them 3 questions. Do they have the same information in terms of pathological content? Which one has better quality? And which one is the real one? This is the question that we are most interested in, of course. And from the results, basically what I will present is based on a consensus. So for the first, it means that at least 2 pathologists have to agree on the answer. And then for questions 2 and 3 is basically the proportion of times that they choose the generated image, right? And our conclusion is that even though the generated images were ranked or rated as lower quality, the pathologists also choose the generated ones as the real ones most of the times, or at least we could fool them. And in terms of staining, there's also a promising result in terms of similarity with the real one. And more important than that, it was consistent among the scores. That is very important, because then we don't have a kind of a bias towards the brown areas. And I would like to do this test with you. I don't know if the screen will help us, but I think this will be fun. So let's see, which one do you think it's the real? Or yes, the real one.
So my logic is, I'm not a pathologist, so I have to use tricks. You told me that the ones that seemed worse quality.
Yeah, you are already biased because I already told you something.
Yeah.
But try to be unbiased, right? Just like from your perception, what would you choose as the real one?
LCB.
B?
B, yeah, B.
Okay.
I think A.
Okay, it's the B. And let's see for this one. Which one would you say? This is already very good, right?
I say B again.
You say B? B, B, B? Okay, you're right. Now.
They can only be right. Not 3 times in a row.
I should put these the other way around. A, very good. And this one, the final one. I love that this is the best reaction, actually. You can decide all the way. So,
No.
no, which one? Well, still, say something. A is the real one?
Yeah, A.
I think
Very good. Yes, the real one is the B. But this really can fool the pathologists. And this is a point, like the real images can also have some artifacts. So it's not only about like looking sharper or, there's a lot of features and a lot of factors that can affect these assessments. But very well done. Nice. And so to conclude, basically, I just want to raise like where we need improvements or where the research should focus attention. So for sure, the loss, and also in pair with that, the metrics, and it can be attached one to another. There's still no way of doing a proper evaluation of the generated images, or even like during training, like a good way to optimize the models. Of course, we should really focus on improving pathology fidelity, and not only like coming up with the realistic images. And then it comes to generalizability. We were using just one dataset. Of course, we want to try two other datasets. It would be better if they don't have those kind of artifacts. For now, that's why we didn't go back to those, because we know that they have flaws. So what's the point? But of course, this is a very important topic. And to wrap up, so this project resulted in 3 publications. The one in the bottom is just a review of all the techniques that have been used. And we also do that first comparison that I told you. We also do that comparison with the 2 public datasets. So it's kind of a benchmarking and people know where the models stand. And then I will present these two works in a workshop in MICAI in September. One is the work that I just showed you. That is this idea of leveraging the adversarial learning. And the other one is a comparison between GANs and diffusion that I didn't talk to you about. And also there's where we published the datasets. And so stay tuned. These are not public yet, but will be in the meantime. And of course, last but not least, I have to thank this team that was behind of this work. It was a very, very good year, I have to say, and a very interesting topic. And I hope you liked it too.